Hi everybody, my name is Thomas Gare. Today is February 19th, 2023, and I will be reporting on hemolytic anemia for my Diseases of the Cell final project presentation. Okay, so anemia can be described by an abnormally low number of red blood cells, also called erythrocytes, in the blood. The word hemolysis or hemolysis literally means the breakdown or rupture of red blood cells. And together, hemolytic anemia is characterized by a low red blood cell count caused specifically by the destruction of red blood cells at a faster rate than new red blood cells can be created. So red blood cells are the cells that are directly affected by hemolytic anemia, but red blood cells are responsible for carrying oxygen to every other cell and tissue in the human body. And so as a result, hemolytic anemia affects every cell and every tissue in the body as a consequence of reducing the amount of oxygen that is carried to these locations. Regarding enzymes, glucose 6-phosphate dehydrogenase or G6PD is most well known for contributing to hemolytic anemia. This enzyme exists in the cytoplasm of all cells, including red blood cells. And in addition to its use in glycolysis to produce energy in the form of ATP and NADH, glucose 6-phosphate dehydrogenase functions to prevent oxidative damage to cells. And since red blood cells specifically carry oxygen, they are particularly vulnerable to damage from reactive oxygen species like hydrogen peroxide. And a deficiency in glucose 6-phosphate dehydrogenase often causes hemolytic anemia, since mature red blood cells lack the capacity to recover from oxidative damage to the cell membrane. And the metabolic pathway most affected by hemolytic anemia is the electron transport chain and oxidative phosphorylation. Oxygen serves as the final electron acceptor in the electron transport chain, which transfers high-energy electrons carried by NADH and FADH2 to oxygen molecules, which are very electronegative, which forms water. And this simultaneously generates an electrochemical gradient of protons across the inner mitochondrial membrane. The potential energy stored in this electrochemical gradient is then used to generate ATP. And without oxygen, the electron carriers can't offload their energy, their high energy electrons, or move protons up the concentration gradient. And as a result, no ATP can be generated by oxidative phosphorylation. So since hemolytic anemia reduces the amount of oxygen available for use in the electron transport chain, this condition results in low energy availability to the cell and to the organism as a whole. And regarding cellular functions, for every cell or for every cycle in the electron transport chain that is coupled with oxidative phosphorylation, around 30 to 32 ATP molecules are generated. For glycolysis, on the other hand, which produces ATP in the absence of oxygen, only 2 ATP molecules are generated. So the lost capacity for ATP generation is the most serious cellular function that is impacted by hemolytic anemia. So regarding impacted organs, every organ in the body is affected by this disease, specifically because every organ in the body needs oxygen. However, one of the primary organs that's affected by hemolytic anemia is probably the spleen. And this is because the spleen is responsible for filtering out damaged red blood cells. Circulating blood passes continuously through structures in the spleen, which are called splenic cords. And these splenic cords possess a large number of macrophages, which destroy red blood cells with physical damage or protruding antibodies. And since hemolytic anemia results in the mass destruction of red blood cells, the spleen is strained by an excess of objects that need to be removed from circulation. So as a result, the spleen oftentimes becomes painful and enlarged as a result of this disease. regarding bodily functions, since hemolytic anemia causes cells to receive lower levels of oxygen, patients typically present with symptoms of fatigue or feelings of tiredness, owing to the fact that the cells can't metabolize a normal amount of energy. As a result, patients can present with tachycardia, which means fast heartbeat, which results in the heart trying to supply enough oxygen to the blood to compensate. Shortness of breath also occurs from low oxygen or low levels of oxygen as well. Patients present with anemia, obviously, which is a low red blood cell count, as well as hemoglobinuria, or hemoglobin in the urine, which results from the excess secretion of hemoglobin produced by the lysis of red blood cells. And lastly, patients present with jaundice, or a yellow coloring of the skin and eyes, which results from the presence of from yellow-orange bilirubin molecules, which is produced from the metabolism of hemoglobin. Regarding treatments for hemolytic anemia, treatments do exist, thankfully, to fight the disorder. Blood transfusions are common to replace red blood cells lost through hemolysis. Glucocorticoid steroid hormones are prescribed to increase the hemoglobin content and a number of red blood cells, and the total number of red blood cells carried in the blood. In more severe cases, a splenectomy surgery or removal of part of the spleen can help to reduce the amount of red blood cells that are being removed from circulation by this organ. And then lastly, immunosuppressant drugs can be prescribed, which reduce the strength of the patient's immune system if the immune system is accidentally targeting healthy red blood cells and tagging them for destruction by macrophages. And dietitians can help patients suffering from hemolytic anemia by making sure that the patient consumes the vitamins and minerals necessary to maximize red blood cell production. Diets for people struggling with hemolytic anemia are individualized, which means that a universal set of guidelines doesn't exist to help people suffering from the disorder. But dietitians can make sure that the patients receive nutrients such as folate or vitamin B9, cobalamin, which is vitamin B12, and iron, which are all needed for the formation of new red blood cells. So, regarding heritability, hemolytic anemia can be either heritable or acquired, or not, which means non-heritable. Abnormalities in red blood cells that occur from inherited hemolytic anemia are grouped into three different classifications. The first one is membranopathies, the second is hemoglobinopathies, and the third is enzymopathies. And the opathy suffix just means a disorder that is relating to the root word. So in inherited hemoglobinopathies, there is a defect in the structure of the hemoglobin molecule, which leads to reduced oxygen transportation. There are more than 600 different known hemoglobin variants that result from this disorder, which can stem from over 1,200 different known alterations in the human genome. Luckily, not everyone possessing this disorder actually shows symptoms, but around 7% of the global population are carriers of hemoglobinopathies, interestingly. So, regarding membranopathies, there's a defect in the structure of the red blood cell membrane. The red blood cell membrane possesses three main components: a lipid bilayer on the outside, a cytoskeleton network on the inside, and linker proteins in between those two layers. Red blood cell membranopathies result from abnormal cytoskeleton proteins, and the outcomes of this include reduced cell surface area, changes in cell shape, and increased sequestration by the spleen, and cell lysis results from anemia as well. Spherocytosis, shown in the picture down at the bottom, is a type of inherited membranopathy that is characterized by spherical or circular red blood cells that have fragile cell membranes. Interestingly, this disorder is autosomal dominant, and elliptocytosis, shown in the bottom picture as well, is a second type of inherited membranopathy that is characterized by elliptical or oval-shaped red blood cells. and this disorder is actually autosomal dominant as well. In inherited enzymopathies, there's a defect in the enzymes involved in glycolysis. ATP is only produced through glycolysis in red blood cells, and as a result, damage to any of the enzymes involved in glycolysis risks affecting the red blood cells' energy balance. And the reduced ability to withstand biological stresses increases the risk of hemolysis, or the rupture of red blood cells. These disorders are almost always autosomal recessive, which is a good thing. Glucose 6-phosphate dehydrogenase deficiency is an X-linked recessive disorder caused by mutations in the glucose 6-phosphate dehydrogenase gene, which negatively affects the stability and the activity of the enzyme. And epigenetic factors can influence the development of hemolytic anemia as well. Epigenetic factors are non-genetic factors that can affect the disorder. Regarding glucose 6-phosphate dehydrogenase deficiency, which is responsible for enzymopathies, non-heritable up and down regulation of the gene responsible for glucose 6-phosphate dehydrogenase can contribute to an increased risk of hemolytic anemia as well. And hemolytic anemia can be acquired in a non-heritable way through autoimmune hemolytic anemia also. In this process, the body mistakenly deploys antibodies against healthy red blood cells, which causes unnecessary erythrocyte destruction, or the destruction of red blood cells. So in addition to antibody or autoantibodies, the body mistakenly deploys macrophages, which are large white blood cells that destroy objects by phagocytosis, T cells, which are white blood cells that destroy the patient's own infected cells, and they also activate other immune cells, and cytokines, which are small proteins that function in cell signaling. And also aging of the patient's immune system is a primary mechanism which is involved in the development of autoimmune hemolytic anemia also. So regarding subsequent generations, certain forms of hemolytic anemia are heritable. Inherited enzymopathies are typically autosomal recessive, which means that both parents must possess a copy of the recessive gene for the child to be affected. Inherited membranopathies are typically autosomal dominant, meaning the child will be affected even if only one parent possesses a copy of the gene. And then lastly, inherited hemoglobinopathies are typically autosomal dominant as well. In children who have inherited in hemolytic anemia from one or both parents, meaning they possess the disorder, symptoms of the disease can appear early in childhood and do not necessarily only show up later in life. In some cases, the patients can appear asymptomatic. However, children can present with the same symptoms as adults, which includes fatigue, pale skin, dizziness, headaches, and shortness of breath. These symptoms can also indicate a number of different ailments, unfortunately. So more specific symptoms, which are more indicative to hemolytic anemia specifically, are jaundice, an enlarged spleen, dark coloring of the urine, and upper abdominal pain. And so hemolytic anemia involves the premature destruction of red blood cells. There is no clear indication that this disease is specifically caused by failures in the cell cycle checkpoints. Instead, hemolytic anemia is caused by mutations in the human genome, or incorrect regulation of gene expression, and lastly, the targeting of healthy red blood cells by misdirected white blood cells. So, factors in disease development. Lifestyle habits such as diet can increase the risk of developing hemolytic anemia. Deficiencies in dietary iron can result in the lack of hemoglobin. And deficiencies in vitamin B12 can result in failed maturation of red blood cells as well. This can also lead to premature death of red blood cells and lysis of red blood cells that grow to be too large, or their growth is uncontrolled. The risk of developing hemolytic anemia increases with certain conditions also. Patients with heart disease, might potentially possess a prosthetic heart valve that contributes to a paravalvular leak, and this just refers to blood that can leak between the natural tissues of the heart and the prosthetic heart valve. And in this case, hemolytic anemia can occur from stress on the red blood cells that are passing through these spaces. In the disease called sickle cell anemia, the body manufactures abnormal hemoglobin molecules, which cause red blood cells to acquire a sickle shape or a C shape. These deformed cells have trouble passing through blood vessels, and they have a shorter lifespan also. They usually only survive 10 to 20 days compared to the 120-day lifespan of a normal red blood cell, and this condition can result in hemolytic anemia also. The disease called thalassemia has a similar outcome. Here, the body fails to produce sufficient quantities and certain types of hemoglobin, which result in a shortage of normal and healthy red blood cells. And again, hemolytic anemia is a consequence. Other factors such as infections can lead to hemolysis and anemia, which can result from malaria and tick-borne diseases. Pharmaceuticals can cause the condition. Drugs such as penicillin, acetaminophen, and unfortunately anti-malarial medicines sometimes can cause immune reactions that can cause damage to red blood cells as well. So associated genes. Hemolytic anemia can be caused by many different factors, which can either be heritable or acquired. In the case of inherited hemolytic anemia, genes are involved in the development of the disease. 75 different genetic loci play a role in genetic mutations which control red blood cell structure and function. All of the third bullet point there, all of those genes are associated with hereditary red blood cell membranopathies. On the fourth bullet point down, those genes are associated with hereditary red blood cell enzymopathies. And the last bullet point at the bottom of this slide, those genes are associated with hereditary red blood cell hemoglobinopathies. And so as you can see, a ton of different genes can contribute to the overall disorder. And the inheritance pattern, location on the chromosome, and sequence of each gene is well known, which is a good thing. Traditionally, diagnosing inherited hemolytic anemia involves analyzing red blood cell morphology, or the size and shape of the red blood cell, measuring red blood cell enzyme levels, and analyzing red blood cell membrane proteins. Unfortunately, these observations can overlap with different unrelated conditions, and so as a result, conventional diagnostic techniques are not always able to clearly isolate hemolytic anemia as the cause of an observed symptom. More reliable diagnostic tests, such as genetic testing, is highly recommended for identifying hemolytic anemia in combination with the traditional techniques. So only two genes responsible for hemolytic anemia, glucose-6-phosphate dehydrogenase glucose-6-phosphate dehydrogenase gene, which is responsible for glucose-6-phosphate dehydrogenase deficiency, and PGK1 gene, which is responsible for phosphoglycerate kinase 1 deficiency, are X-linked recessive traits, which means they're sex-linked. The rest of the genes responsible for hemolytic anemia exhibit a mix of autosomal dominant and autosomal recessive inheritance patterns. So for diagnosis methods, genetic onset of hemolytic anemia is caused by mutations in particular genes. And since so many different genes can contribute to the development of the disease, different types of mutations have been identified. In hereditary red blood cell membranopathies, nonsense and frameshift mutations are most common. Nonsense mutations involve the addition of a premature stop signal. A premature stop signal, which causes translation of a gene to stop too early. And a frameshift mutation involves the addition or deletion of nucleotides in a gene, which shifts the entire reading frame and causes a dramatically altered protein translation. In hereditary red blood cell enzymopathies, missense mutations are commonly identified. This involves a change in single cell or a single nucleotide in a gene, which can cause an correct amino acid to be substituted for an incorrect amino acid. And then in hereditary red blood cell hemoglobinopathies, deletions often occur, which involves the loss of nucleotides from a gene. And once again, this results in a frame shift mutation, which shifts the entire reading frame and dramatically alters the final protein translation. So, continuing, for diagnosis methods, direct antiglobulin test, abbreviated DAT, is the most effective way to diagnose autoimmune hemolytic anemia. Autoimmune hemolytic anemia often occurs when red blood cells are attacked by autoantibodies, or antibodies that target structures that belong to the individual's own body and should not be attacked. These autoantibodies are often immunoglobulin G, or IgG. The DAT test can often determine whether individuals or whether immunoglobulin G antibodies are bound to red blood cells. This can be accomplished by adding a substance to a blood sample that clumps antibody-coated cells together. If the cells do clump together, they're likely coated with antibodies, which indicates autoimmune hemolytic anemia. And if they do not clump together, autoimmune hemolytic anemia is unlikely. And you can see in the picture on the bottom of the slide there a clumped blood sample versus a non-clumped blood sample. And the clumped indicates autoimmune hemolytic anemia, whereas the non-clumped sample does not indicate autoimmune hemolytic anemia. Several different techniques do exist to identify acquired hemolytic anemia. First, a simple blood smear viewed under a microscope can detect defects in the red blood cell membrane. An increased number of reticulocytes or immature red blood cells can indicate that bone marrow is attempting to replace a rapidly decreasing number of mature red blood cells and is compensating by trying to grow a large number of new or immature red blood cells. Lactate dehydrogenase is found in high concentrations in red blood cells, and as a result, increased number of this enzyme in the blood can indicate abnormally high rates of hemolysis or destruction of red blood cells. And then lastly, haptoglobin is a protein that's made in the liver, which binds to hemoglobin after being released from red blood cells. And in hemolytic anemia, haptoglobin binds to excess free hemoglobin faster than the liver can produce new unbound haptoglobin. And so as a result, low haptoglobin levels can be an indication or a sign of hemolytic anemia. And all of these tests can be completed with laboratory testing. Some of the more specialized tests, like the DAT test, aren't a part of routine blood work, but they can be ordered by a primary care doctor and do not need to be ordered by a specialist. And regarding preventative measures, genetically acquired hemolytic anemia unfortunately cannot be prevented. For acquired hemolytic anemia, preventative techniques do exist. Aside from avoiding certain pharmaceuticals, which can initiate immune reactions that damage blood cells or damage red blood cells specifically, vitamin E has been shown to make a difference in red blood cell stability. The vitamin can prevent oxidation of fatty acids in the cell membrane of red blood cells, and as a result, the membrane is less fragile and more resistant to lysis. Vitamin E has also been shown to accelerate erythropoiesis, or the production of new red blood cells. So for treatments thankfully do exist to help fight the disease. Blood transfusions are common to replace red blood cells lost through hemolysis. Glucocorticoid steroid hormones are prescribed to increase hemoglobin content and the number of red blood cells in the blood. In more severe cases, a partial splenectomy surgery or removal of the spleen can help to remove or can help to reduce the amount of red blood cells that are removed by the spleen. And lastly, immunosuppressant drugs, which are often corticosteroids, are prescribed to reduce the strength of the patient's immune system if the immune system is incorrectly targeting healthy red blood cells and tagging them for destruction unnecessarily. And each of these treatments is an established technique for treating hemolytic anemia, and it's proven to be successful. So for the outlook of the disease, the research frontier involving hemolytic anemia primarily surrounds autoimmune varieties of the disease. All the treatments that I just described are effective in treating acquired hemolytic anemia. But for autoimmune hemolytic anemia, active research is still being conducted on manipulating the immune system to eliminate negative effects on red blood cells. And actually different pharmaceuticals or different drugs are being experimented with to accomplish the goal. So the primary focus of these drugs are B lymphocytes, which are white blood cells that produce antibodies, and T lymphocytes, which kill cells that are tagged with certain antigens. A receptor on the cell membranes of T lymphocytes called interleukin-2 receptor, abbreviated IL-2R on the second bullet point there, is a target for the new experimental drugs. A medication called sirolimus binds to the interleukin-2 receptor, and it disrupts the function of T lymphocytes that have lost normal function and have started to incorrectly destroy healthy red blood cells. And a medication called idelalisib has the same effect on faulty B lymphocytes. An important observation has been that in children recovering from kidney transplants who suffer from autoimmune hemolytic anemia as a reaction to receiving the new organ, sirolimus has successfully managed the patient's hemolysis symptoms. And in addition to all the old well-established treatments that are available, the new medications thankfully offer a novel strategy to help the autoimmune varieties of the disease moving forward. And although the current survival rate of autoimmune hemolytic anemia is already fairly good, at over 82%, these new experimental drugs have the potential to make that number go even higher. And here are my references for the presentation. Thanks, everybody.